Okay, looks like everybody jumped out to grab something to drink. Everybody can come back in here when they get a chance. We'll get started with our second speaker here. So it's my pleasure to introduce Dr. Francelino Rodrigues. He comes to us from Mexico at the International Center for Maize and Wheat Improvement, CIMMYT. And he works with the Biometrics and Statistics Unit, but his specialty is actually in precision agriculture. And he's going to talk to us today about using precision ag and remote sensing techniques to make genotype selection in a breeding experiment and the data analysis and tools that go along with that. So with that, let's welcome Dr. Rodrigues.
Well, good morning, everyone. Thanks a lot for the organized committee to invite me to come over here. For me, it's a pleasure to come and have this opportunity to share with you guys a little bit what we are doing in CIMMYT. Well, these spots over here, over this map, are all CIMMYT stations across some countries. Okay, we have a little bit about the reality of CIMMYT nowadays. We have offices across 14 different countries. They are in Africa, located in Ethiopia, Kenya, and Zimbabwe. In Americas, we have in Colombia and Mexico, where is our headquarters. And in Asia, we have offices in Turkey, Iran, Kazakhstan, Pakistan, Afghanistan, India, Nepal, Bangladesh, and China. Well, CIMMYT is divided by some research programs. In total, it's 5 different research programs. We have the Global Conservation Agriculture Program, the Global Maize Program, the Global Wheat Program, the Socioeconomics Program, and also the Genetic Resource Program. My unit, the Biometric and Statistics Unit, is located into the Genetic and Resource Program. It's a support unit, which the idea is to give support to all over across these programs in everything about statistics, and now that I'm there, everything about proximal and remote sensing, the use of those sensors and how to deal with this data. CIMMYT now has more than 200 scientists, and in total staffs is like 1,100 staffs across all the offices. And in Mexico, we have more than 700 staffs in Mexico. In scientists, it's about 85 to 100 scientists in our headquarters. Well, this is literally an overview of our headquarters. This pic was taken with a UAV flying above ground, more or less 190 meters above ground, and we yielded, in this case, 5 centimeters of pixel resolution. Our headquarters has an area of 93 hectares, and in this case, it was an RGB and near-infrared sensors that it was on a UAV. Well, the contents of my presentation, I'll be giving an overview about the technologies in use at CIMMYT for field characterization. Inside this, the ground-based sensors, the remote sensing platforms, and then I'm going to show a studied case from last year, going through the exploratory analysis, the methods used for the data analysis, and some brief results from this case. Okay. Well, Tabari, this morning, he justified my work, actually. He said about the cost of phenotyping. And actually, the idea of this is to decrease cost of phenotyping. So we have here cost of breeding to achieve better varieties, cost of breeding, phenotyping, and molecular. So the goal is to decrease the cost of this guy here. Well, going for some technologies. The technologies that it's in use at CIMMYT for field characterization. We have been working with the spectral radiometer, with IR thermometer, green seeker, and some scientists are getting some good correlations, for instance, with canopy temperature and grain yield. Here we have some examples of biomass, predicting biomass with NDVI, senescence, plant water status, working with canopy temperature, grain yield prediction, working also with NDVI and canopy temperature. Well, the soil sensors that we have there, we have the GPR, the ground penetration radar, which actually this guy is a PhD candidate from Texas A&M. He was doing his PhD in partnership with CIMMYT, and the goal of his work was to estimate root using the ground penetration radar. Okay, and here we have the M38, which, well, people from the precision ag area, it's really familiar with this sensor. It's a sensor which measures the apparent electrical conductivity of the soil, and this is measured through electromagnetic induction. So we go through the field, capture measurements by second. In this case here, like it's getting through a sled. So we have here mounted in a sled. We have the RTK system, so it's all connected. And we go through the field by 10 kilometers per hour and getting measurements in 1 hertz. So each second we're getting measurements of the soil. This is for field characterization. Okay, the remote sensing platforms that we have in CIMMYT. We have a physiologist from the WIT program, Maria Tatares. She's working with these 2 UAVs, which one is a helium-filled blimp with payload of 6 kilograms, and another one is this helicopter, which the brand is Aztec Falcon. The payload is much lesser; it's like 650 grams. So both of them are carrying different sensors. In the case of this, the helium blimp can carry it's carrying a tetracon with 12 channels with 1.3 megapixels resolution, and the spectral range from 445 to 980 nanometers, split into 12 channels. The Aztec Falcon carries a tetracon CIR, which is just the RGB and near infrared, and also a FLIR camera, which is a thermal camera, and a normal RGB camera, which is just a Sony with 40 megapixels. And we also are dealing with the airborne remote sensing platform, which we have these 3 different sensors. We are dealing with the tetracon with 6 bands, the thermal camera, and also with a hyperspectral camera. These all we put by couple, like the hyperspectral and the thermal, or the multispectral and the thermal. Here is a photo from the hyperspectral sensor, so it's really compact. And just beside here, there's a hole that we put the thermal camera as well. Well, going for the studied case, predicting yield through proximal and remote sensing data. So this is an image from our station in Obregón, north of Mexico. This image, it's a multispectral image. We took this image flying 300 meters above ground with the airborne platform. Here it's a representation of the image using on the red band 800 nanometers, on the green band 670 nanometers, and on the blue band 550 50 nanometers. Flying 300 meters above ground, this gave to us, we yielded this mosaic with more or less 30 centimeters pixel resolution. These two tiny yellow things are the experiments that I will be showing the data analysis, okay? So, and actually the area of the experiment is like a 0.1 hectare, a little bit less than this. So, actually it's something that it's a shame. I'm sorry, but I didn't calculate the full area of the mosaic. But you guys can have an idea about according with the experimental area. Zoom in on the experiment. So we had two experiments, a full irrigated, which you called FIG, and a reduced irrigated, which you called REED. Okay, so they were sowed on November and December 2012. The harvest was on 15 May 2013. Flowering stage, more or less, in average, February, March 2013. So we have the REED, 163, 164. In each of them, they are split. They are in total 192 observations, split into 3 reps. And inside each rep, we have 8 checks, which are all the same, plus 56 genotypes, different genotypes. So we have 56 genotypes here, more 56 genotypes here, and those are the same on here, on this experiment, okay? During the season, we flow the area, so we got 6 multispectral plus 6 thermal images with the airborne platform, with the description that I said before, flying 300 meters above ground and 30 centimeters resolution. We flow from 14 March to 24 April. And we also took EM38 survey in horizontal and vertical dipole. Just to clarify, horizontal and vertical dipole is like more shallow and more deep. Like we can, the attenuation of this is like 75 centimeters on the horizontal, and on vertical 1.5 meters. Well, about the vegetative index. So we have these 6 bands. We have the 550, the 670, the 700, 710, 750, and 800 nanometers. We have these 6 bands. So what kind of vegetative index that we can be calculating with this? So we calculated chlorophyll index, which it's Emicari, Tecari, all those ones. A structural index of the plant. We did red edge ratios index and also RGB index. In total, we generated 23 different vegetative index per image. Okay, so all those images are related with structural, the structure of the plant, the chlorophyll of the plant, and some red edge and RGB. Well, the data we have. So we have 2 REED experiments plus 2 FIG experiments in a total of 112 genotypes plus check in a completely randomized design with 3 replications. And we were working with yield. We want to predict yield. Okay. Inside this, we have the soil apparent electrical conductivity, EC, in 2 depths. And we have the 23 different vegetative index plus thermal information times 6 images across the crop stage. Well, data analysis, we calculated all that vegetative index. The exploratory analysis was based on to check the behavior of the vegetative index across time and check the correlation of the vegetative index with the checks, doing agile statistics of the EC information to check the spatial variability of the EC across the experiment, across the plots, adjusted the means of yield by genotype using restricted maximum likelihood, and then going for the modeling and validating. So yield by predicting variables. Okay? Okay, so some results for different approaches. The exploratory analysis, where I said before, checking the VIs, the checks, and the EC. It's a box plot, like where we have here the image. So this is the first image, this is the last image, okay, for each of the vegetative indexes. And using, like checking the response of all the genotypes across this. So we have here the temperature, the light green, we have the chlorophyll index, the dark green is we have here the structural index, we have on the red HO index, and here the RGB index, okay? So we can see like the range variation across all the genotypes for each of the vegetative index, and we also can see some temporal pattern. We can see some temporal pattern for some of the vegetative index, okay? And in some of the images, we also see like a really big range of response according with different genotypes. This is the data of the read experiment, just the read experiments, okay? Both with 112 different genotypes. And in this case, it's the FIG experiment. We can check the temperature and also, again, the chlorophyll index, here, the structural index, the radial, and red edge ratio, and then the RGB index. We can see that in some of the vegetative index, this range variation across genotypes, it's a little bit bigger than the previous one. But we also can see the temporal pattern. Okay, so again, it's the same. First image here, last image across time, across the crop cycle. We can check that there is some temporal pattern according with different vegetative indexes. Here are some correlations with checks. So the correlations of the read checks and the FIG checks. We calculated this correlation just to have an idea about which of the vegetative index, which of our variables can be explaining more what we want to predict, which is yield. Okay, so it's yield from the checks and the vegetative index. In the case of the read, the temperature was the most important variables. And in the case of the FIG, the T-carry and the M-carry vegetative index were the most important in this case. The correlations are on decreasing, okay? And we can see that it varied, it ranged from 0.73 positive to minus 0.8. And in the case of the read, and in the case of the FIG, ranged from here 0.6 to minus 0.7. So, but this is checks, okay? This is not anything about the genotype set. Here there's just statistics analysis with the EC information. So across the experiment, this is the read experiment, okay? So we have the post-plot information which we divided through quantiles to check if it does exist some spatial trend across the experiment. This is one depth, okay? This first column is the vertical dipole. This second column is the horizontal dipole. Then we went for variogram, plotting a variogram in 4 different directions to check if it does exist some anisotropy on our data. And finally, we choose for the isotropy variogram. So we fitted a theoretical model, and then going for the Kriging And the cross-validation, we check like on the vertical dipole, we got 0.7 R squared, and on the horizontal dipole, we got 0.6 R squared of cross-validation, okay? And for the fig, we check, again, the post plots for both depths. So we check here that just with the post plot, we can check that it does exist some spatial dependence of the data. Here, plotting in different directions to check the anisotropy. In here again. And then fitting the theoretical model to predict and to do the kriging. This is the vertical information, which we got 0.84 of cross-validation. And this is the horizontal dipole, which we got 0.63 of cross-validation. And here are the maps of the spatial variability of EC across the experiment, where we have the horizontal and the vertical dipole. So we can check that it does exist some spatial variability of the soil inside this, even that the range variation is not that bigger, okay? Here is for the fig experiment, and here for the reed experiment. Again, horizontal dipole, vertical dipole, and the spatial variation of the EC across the experiment. Here in the case of the reed, we got a little bit less range variation than with the fig. This is to have an idea about the experiment by itself, like do we have soil variation across the experiment or not, just to have an idea about this. And just to clarify, the AM38, the most important factors that influence on the response of the AM38 is salinity, pH, clay content, and moisture. So it's a metric, it's not straightforward. So there are a lot of factors that can be influenced on the response of this sensor. But it gives an idea about the soil texture, okay? Okay, well, going for the data adjustment, we did the data adjustment using restricted maximum likelihood. And okay, so in the case of the fig 6.3 and fig 6.4, using rep plus genotype as fixed effect, we got satisfactory results on the f-test. So here the rep p-value and the genotype p-value. So our rep were not significant. Our genotype, yes, it was. In the case of the reed, it was a little bit more, we need to try a little bit more. So the first tentative with the rep plus genotype as fixed effect, we saw like rep as significant, genotype as non-significant. Then what we did, we added the coordinates of this as random effect. So then we changed a bit the p-value of the reps. So this means that, okay, it might exist some trend across the experiment, okay? But it's still not sufficient. And then we adjust the interaction of the coordinates into an autoregressive model. And we got satisfactory results of the f-test for the rep and for the genotype. So this means that it doesn't exist some trend across the experiment, okay? And in the case of the 164, first trying just rep plus genotype as fixed effect, all significant. Going further on this, adding just interaction of the coordinates into autoregressive model. We got some change of the p-value of the reps. And going further on this, the interaction of the coordinates plus the plot numbers into autoregressive model as random effect, okay? Still, the rep and the genotype as fixed effect. Here, then we got satisfactory results for the reps and for the genotypes. So we adjusted the means So now what do we have? We have 130 observations, adjusted means of each genotype plus the checks against 146 predicting variables across time. So why 146? We have 33 vegetative index plus thermal information for each image times 6 plus the two information of the soil, the two depths of electromagnetic induction. So definitely we have an overfit, okay? So how to turn on this? Turning. We used some approach on selecting variables, multiple regression, PCA, PLS, bootstrapping. Any statistics tools can be used on this. It's a matter of going and test different ways and check how you can be describing this, how you can reach your goal. One interesting approach that discussing with the team and we got on this, like the first question, how to decrease this data set, okay? So we were curious to check the genotype behaviors across time, okay? This for each vegetative indexes. So here for the read, here is a similar graph of the box plots, but we have each line representing one genotype, okay? So we have temperature, we have again, the chlorophyll index, the structural index, red edge, and RGB index, okay? Images across time. So each line, it's representing one genotype. So we can see like how huge is the range variation of each genotype inside each vegetative index, okay? And we can see how this can change across time as well. So this is a plot of using the smooth effect. We just linked the points across time to check the behavior, the real behavior of these genotypes for each of these vegetative index, okay? And here, first read, okay guys? And here, the quadratic effect. So we had that smoothed fitting, giving us an idea about the range variation of the genotypes for each vegetative index. So, and here we did a fitting with a quadratic effect, with a quadratic effect, okay? Checking if we can capture this trend across time without losing information. So we can see that we can say that it's quite similar about trend across time, this fitting. And here, just a linear effect fitting. So for each of the genotypes, again, for each of the vegetative index across time, we can see that we capture some main trend, temporal trend across time, but in the middle, we can be losing some information. So in this case, we will be losing some information, okay? And these are the same graphs for the FIG experiment. So here, again, temperature, all the other vegetative index, and we can check the range variation of some different genotypes, okay? It's some really big range. And also, what is cool is like for some specific stage of the crop, this range can be higher, and for other stages, it can be lower, okay? This, again, the quadratic effect fitting. So we are catching some. It's not that different from the previous one, so we can capture some trend across time. And here, the linear effect, okay, the linear effect fitting. So what did you get from this? So we fit each genotype by vegetative index, okay, grabbing the fit coefficients. So we grab the intercept, the linear, and the quadratic effect of each fitting, okay? So in this case, what we got in the linear effect, considering the linear effect of each genotype, we had 130 observations against 50 predicting variables. So we decreased it from 146 predicting variables to 50 predicting variables. And on the case of the quadratic effect, which describes a little bit better the temporal variation of the genotype's behavior, we decreased it from 146 variables to 74 predicting variables. Okay, so that was the data set that we used for going to the modeling process. Okay, on the modeling process, just warm up a bit. Multiple linear regression using the 100% of the data set and using the 50 predicting variables of the linear effect. Okay. In the case of the read, we got adjusted R-squared of 0.57 and p-value considerable satisfactory. And in the case of the fig, we got 0.56 R-squared adjusted, statistically significant, the model. But still, we are dealing with 100% of the data set and all the 50 predicting variables, okay? So we choose to go for a stepwise model, a simple stepwise model to drop some of these variables in a backward and forward direction, and choosing as criterion the archaic index. So again, on the 100 data set, using the 50 predicting variables. So we drop for 30 variables in the case of the read. In the case of the fig, we drop for 27 variables, increasing our power of adjustment to 0.61 in the case of the read and 0.63 in the case of the fig. Again, we asked ourselves, how reliable is this? How reliable is this fitting? How this can be replicated? Okay, so we did this process inside a bootstrapping, so in this case, we did 100% of the data set inside the bootstrapping, which is a resampling method, okay, with 1,000 interactions. In the case of the read, well, as output of the bootstrapping, we have this density graph. It's a shame that I didn't increase the font, sorry. We have this density graph and we have the histogram graph of the R-squared adjusted. Okay, so inside the same data set, how this can vary, how this adjustment can vary inside the same data set. Okay, so here we have like as mean I think 0.65 in the case of the read, and in the case of the fig, we have here 0.7, more or less, the average, okay? And we decided to split this data set, okay? So we split this data set in 70%, randomly in 70% to fit the model, and then we applied this model on the rest of the 30% to check how is our power of prediction. So we split this randomly, we fitted the stepwise model, and we applied this model into a validation, into a bootstrapping, using 5,000 interactions, okay? So as output of this, we have, again, the density graph and the histogram graph. In red is the correlation of the measured yield and the predicted yield. And here in dark color is the R-squared of this. And here in gray we have the correlation again, and in blue the R-squared of the predicted yield and the measured yield, okay? So we can see that this output is from the 30%, okay? So using the model that we fitted, applying on this 30% into a bootstrapping. So we got around 0.6 for the read as average. And for the fig, we got around 0.5. Actually, to be honest, I'm not seeing much. Actually, I remember in the top of my mind these numbers, okay? Okay, so it's still like how to explore a little bit more this data. Then we used the same approach before, splitting the data set, but in a subset regression fitting. And the subset, so we applied the subset regression fitting on the 70%, fitting this, and then we got this model, and we applied on the rest of the 30% into, again, a bootstrapping with 10,000 interactions. The good point of this subset regression is that you can set the maximum number of variables that you want to get into the model. So we choose by purpose, like I want a maximum 6 variables, because it could happen that the intercept of one vegetative index get into and the linear effect of other vegetative index get into the model. In the state of art of remote sensing, this doesn't make sense. So the idea is to fit a full model using a full vegetative index information across time. So we set up like as 6, think that we can go up to 12 predicting variables, okay? So as output, we have this maximum of 0.59 of R adjusted using these specific variables. But then each of those that get into there, if the intercept of RDVI and the linear effect of the TVI get into, we added the full vegetative index information across time, okay? So this is the density graph, again, with the correlation with the R squared, which on a cross-validation we got around 0.665. The histogram, which we can visualize better, the correlation of predicted yield and measured yield, okay? We got, again, around 65, sorry. This is for the read model. And for the fig, this is the same graph before, so we can check which are the variables that we can choose. And here, the density graph with the correlation and R squared again, and the histogram graph. So in the case of the linear, the linear effect, the linear data set, the models that we select to use the temperature, TVI, RDVI, and the ratios of these wavelengths. Well, then we were curious to apply the quadratic effect coefficients. So to use the quadratic effect data set to check if you can get better prediction. And we applied it the same form that we did previously. So 70% of the data set, fitting, applying only 30% as a cross-validation into bootstrapping. Okay? So again, here is just the graphs of the outputs. Here we have the density graphs. And here we have the histogram of prediction, which we reached, it's 0.6 on the read, and it's 0.7 more or less on the read, and 0.6 on the fig of correlation between measured yield and predicted yield by the models. Okay, and in these models used RDVI, NDVI, and T-carry by Osavi vegetative index, and Osavi, MCARI, GM1, MCARI by Osavi vegetative index. So then we asked, okay, our quadratic effect model for the read, we used RDVI and we used NDVI index and T-carry and Osavi. And for the fig, again, the Osavi, MCARI, GM1, MCARI, and Osavi. We can check that some of them maybe telling the same thing. So they have the same behavior across time. So we might be able to drop out one of these without losing power of prediction. In the case of the fig, they have Osabi and GM1 telling with a similar behavior, which, again, we may be able to take off one of those without losing power of prediction. And the idea is, like, now, does it make sense? That's the question. We are dealing with a bunch of numbers and vegetative index, which is truly telling something about the plant. And what we want is to have some model that we can explain what is happening there. So we can say in the end that, okay, you can deal with 3 specific vegetative index, and you can be predicting in a certain percentage of, you can be predicting yield with a certain percentage of power. We are still, the step of this now, we are still working on this, if does it make sense or not. And we are trying another tools, all those who are linear models, we are trying non-linear models now, and we are checking with other experiments if how reliable is this, how replicable is this vegetative index for predicting yield, and if does it make sense or not. Okay? That's the end. Well, I take home message. Statistics is a freestyle. So many different tools may be used to solve the same problem. This is for sure. My acknowledgements. I would like to say thank you to the BSU team, and specifically to Zakaria Kelwell, which is a biometrician over there, the Global Conservation Ag Program, the Global Wheat, and the Global Meat Maize Program. To Maria Tataris and Jill Keynes, which are 2 physiologists from each of those programs. And Fernando Toledo, which is a PhD candidate from Brazil. Well, it would be this. Muito obrigado. Thank you very much.
So we have some time here for any questions for Dr. Rodrigues, if anybody's interested in.
Interesting presentation. What is the general approach you think moving forward of this data analysis when you have this VI, so many VIs? I'm looking at your presentation. You have presented R and R square, and then you're looking at the center point of the mean or the medians. Those smaller R values, some of the iterations was just because your data set is small that you can have those extreme cases where the prediction is really bad, or what are the, your general thoughts on moving forward with this data set?
Yeah, well, we are trying now to understand, and actually we are trying to replicate this kind of data analysis into different data sets, because actually this is just one example. It was the first year of flying, and actually the experiment wasn't designed for the images. So it's one thing that we are now, the idea is to move forward, like to set up experiments that we know that we will be flying, insert crop stage, and then move forward for this, from this data analysis, like into, we can be testing like non-linear models as well, and check the power of this prediction. How can we be predicting? Yeah.
Another question. We were talking with some of the crop modelers, and then once you transform those curves for each genotype, and they'd like to take those parameters, the linear function, a quadratic function, those intercept, and then those 3 terms, as an intrinsic property of each genotype. And then you could actually try to do genetic mappings of those coefficients. What do you think about that approach?
Oh, it could be done, I think, yeah. And actually, Like we tried also, I didn't put here, but actually we tried so many things. PCA, PLS, like, because we need to be using this temporal information. I think this is information that we cannot be losing. And the idea is like, no, for sure, like you can be feeding this, you can grab this. Actually, in this case, we grabbed the information of each genotype. And then we went for the modeling. Yeah. I don't know if I answered your question, sorry.
So are there any other questions from the audience?
Questions are good, people.
Nice presentation. I just had a quick question. Can you explain a little bit about the breeding application of this approach? Is the plan to
like the idea
trash combines and use?
The idea is like if we can tell the breeder that okay, you don't need to harvest your whole plot. You can harvest like 2/3 of your experiment or 1/3 of your experiment, and we can predict the rest. So like laboring is something that for harvesting, for all these things, it's something that we can be applying for this.
Okay,
the major idea, like...
Well, one idea would be then to just save labor from combining.
Yeah, it's one idea. Sure, it's one idea. Hmm? Definitely. Yeah.
Are there any other questions? No, I have a question. So you said that this experiment wasn't designed to be flown over. How would you have changed it? What's the specific setup of an experiment that you would use to have this aerial platform?
I would maybe dealing with more genotypes numbers, like increasing the number of genotypes and a little bit the numbers of replications. So in the end, when we adjusted the means, we are able to have more observations. And then we can also play like of not using all the reps, like to adjust the means, to be able to go and predict the ones that we didn't use. So I believe that this might be changing. Yeah, sure. And also one thing that, one question that we still don't have is like which crop stage we should be flying. We still don't have this answer. Like is it GS31, is it flowering? We still don't know this. It's something that we need to figure out. Like you can be flying the whole crop cycle weekly, but the bunch of data that will generate on this, like it's huge, and sometimes you do not need. So like if we specify the crop stage that it's better for predicting yield or I don't know, chlorophyll content in the end of the crop cycle, you can, for instance, like with 3 flights, to have the same power of fit.
Are there any other questions? If not, let's thank Dr. Rodrigues.
Thank you very much.